So why do we use mice in neuroscience research? First of all, we have to realize as we do that mice are not humans, very different. However, there are similarities in the processes that go on in a mouse brain and also in a human brain. For example, you can think of a skateboard and a car. There are some things that are shared and similar between a skateboard and a car. For example, the wheels, the axle, different parts like that. And there are parts that are unique to a car which a skateboard doesn't have. Your air conditioning, your radio, your roll-up windows or power windows these days. So what we can do with mice is to look at the things that are common between the skateboard and the car, or the mouse and the human, and the processes that are common between them. For example, in the mouse brain, we can look at how during development the different cells in the brain, the neurons, are connecting to one another. Some of those processes are the same in the mouse as they are in the human. So what is a knockout mouse? A knockout mouse is a mouse which we take a specific gene, which we knock out, meaning we take it out of the genome, and see what's going on with the mouse when that gene is removed. An analogy to that is if you took a lot of different cogs, a lot of different gears, and you pulled one out of a whole system of them. And then now you're going to observe and see how that system functions without that specific gene or that specific cog or gear. Now, when we do that, we can look into the mouse's brain using imaging, we can look at the cells, we can look at the behavior and see, okay, without this specific piece, how does the mouse behave? How are different brain volumes changing? And try to understand how that specific piece contributes to the overall function of the whole system. So what we're realizing now in science is that with a knockout mouse, one specific gene that we're taking out may not just lead to autism, but other neurodevelopmental disorders as well. For example, we can take a specific gene that's found out by other researchers in the PON network, and we're seeing that that gene is not only affected in ASDs, but also in schizophrenia. Using knockout animals, what we're trying to understand is not how that specific gene affects autism, but how that specific gene affects the things that are going on in the brain that will help us understand many disorders, many different diagnostic labels, and not just one specific one. And this applies with autism, with OCD, and many other of the disorders that we're looking at that happen during development. And with these, once we understand these different mechanisms that are changed in these animals, we can then go back into the machinery and try to see if we can target specific parts with different therapies and interventions, and also at very specific time points, since we know this is developmental in nature, that there are perhaps critical windows where one therapy or one intervention may help put the brain on a healthy trajectory rather than moving it later where it's less effective, and understanding how that can influence both our treatments and also how we can influence policy doing that.